everyone. My name is Rapeepan Kongnual from Prince of Songkla University, Thailand. Okay, today I would like to present my research work in a topic, the distinct behavioral response of the chocolate and morphine-induced condition peak preference in mice. Okay, first of all, I would like to introduce everyone about the obesity. The obesity is considered as a public health issue in worldwide in nowadays. And this is a key important and key factor that related with a chronic disease, including the diabetes, the cardiovascular disease, and also the cancer. The obesity can be influenced by the many factors such as the genetic background, the mental health, and also some food can trigger the addiction process, similar with the addictive drug on the reward system in the brain. And the foods that trigger the reward system we call that is a palatable food reward. And chocolate is one of the highly palatable food reward that's used in the reward research. From the previous literature, from the previous study, they comment that the chocolate can arouse the present feeling and reduce the tension and improve the mood. And the chocolate, it is the unique brain activity that seems to provoke the addictive-like behavior process. And morphine is a standard drug that's used for treatment for a chronic and severe pain. That's used in both of cancer and non-cancer patients and in hospitals. And the morphine acts via the mu opioid receptor in ventral tegmental area or VTA brain area that activate the dopamine release in the reward system, and that's induce the addictive behavior. And morphine is used in this research as a positive control. And learning and memory pathway is an important pathway that related to the addictive behavior. The basic experiment that related to the learning and memory pathway that everyone may know that is a classical conditioning or Pavlov conditioning. This experiment is about the response of the dog, that they no respond to the ring sound and they respond to the food. And during the condition, they pair the ring sound with a food and perform with a dog. And finally, the dog respond to the ring sound. And from these experiments, they conclude that the environmental cue can drive the animal to perform the learned behavior. And this conditioning is a principle of the conditioners peak preference technique or CPP technique that's used in this research. And what is the CPP or condition peak preference? This technique is used to examine the reward property of the addictive substance in the animal model. These techniques determine the reward effect associated with the environmental cue. As you see in the figure in the left side, the environmental was divided into three types of the environmental. The first is the CS plus side, that's paired with a stimulant or stimuli. And the opposite side is the CS minus, that's unpaired with the stimuli. And the middle zone, that is a neutral side. Okay, we come to our work, that's the objective. This study aimed to investigate the rewarding effects of chocolate in comparison to the morphine by using the condition peak preference or CPP model in male sweet albino ICR mice. Okay, for the briefly of the methods, I will talk in this slide, and for the detail, I will talk in the next slide. Okay, for the animals were divided into three groups of animals. The first is the control group that represent the normal animals. In this group, animals will receive a saline injection with a food, and the second is an additive drug. In this is a morphine group. The animal were given a morphine injection with a food pellet. And the last one, it represents the food addiction. This is a chocolate group. In this group, mice were given a saline injection and chocolate. And for the busy schedule of the CPP procedure, that start with the neophobia prevention. And next is the preconditioning, and follow with 10 days of the conditioning. In the conditioning period, in day 2, 4, 6, 8, and 10, mice were given a conditional stimuli as I mentioned in above before, and follow with the post-conditioning test. And next is the 10 day of extinctions and extinction test, and the last one is the reinstatement test. Okay, for the detail of each process, I will talk this. Okay, start with the preconditioning. The preconditioning, the mice were allowed to habituate with the CPP chamber. They are allowed to explore chamber freely for 15 minutes for 3 days, and the video of the behavior were recorded on day 3. And the next is the conditioning period. This consists of 10 days. Okay, on day 2, 4, 6, 8, and 10, mice were given conditional stimuli, including the control given saline injection and food pellet. Morphine were given a morphine injection and food pellet, and the last one, chocolate, were given chocolate and saline injection. And on this day, the mice were confined in the CS part of the chamber. And the opposite side, on the day 1, 3, 5, 7, and 9, all mice were received a saline injection and food pellet at the same, and confine them in the CS- side of the chamber. And follow with post-conditioning after 10 days of condition. The experiment were performed as same as the pre-conditioning and record the video. And the next is the extinction period. The next extinction period is quite similar to the condition, but it's different in all mice will receive a saline injection and food pellet every day. On day 2, 4, 6, 8, and 10, mice were confined in CS plus site. And on day 1, 3, 5, 7, and 9, mice were confined in the CS- site. Okay, for the extinction period. After extinction period is extinction test. All mice were given a same thing. Same thing. That's it. A saline injection and food pellet, and allow them to explore the chamber freely as the P condition. And the next one, the last one, is the reinstatement test. Before let them to explore the chamber, the animal were given conditional stimuli. The control group was given a saline injection and food pellet. Morphine was given morphine and food pellet. And the last one, the chocolate group was given a chocolate and saline injection. Okay, for the result, the data were normalized with the preconditioning and displayed as a distinct from the preconditioning and show only the post-condition, extinction test, and reinstatement. Okay, become to my result. You will saw in the heat map in the left side that start with the chocolate. Sorry, that start with a chocolate, morphine, and control. And you will saw that only reinstatement in the chocolate that show an obvious time preference in the yellow scale that indicated the high time preference in this chamber. And you will saw that's only morphine in reinstatement that show an obvious time preference in CS plus site and stem. But in another, that's quite difficult to distinguish them by using the visual. So the CP score was calculated to distinguish the result. And only the CS plus that show the significant difference between only in the reinstatement period. That's compared between the chocolate and morphine and control. Okay, for the next, result is the number of NT, or the number that animal visits in each arm. The number of NT were counted after animal crossed into the arm, and the y-axis that represent the number of entry, and the x-axis represent each group of animals. That's only the morphine that show a significant increase in extinction, but that not show in chocolate or control. And the last result is a locomotor or total distance during 15 minutes of the video. We found that from the total locomotor, the total distance is not significant for all of the experimental group, including morphine, chocolate, and control. But you saw in the right side of the figure, only the morphine that show a repeat tagging line, only CS plus and stem. But in chocolate, they show a stable tagging line that's not change the line rate of the movements. Okay. For this result, I discusses the effects of portable food on the viewer's behavior that depends on the various factors. The first thing that might be the energy balance, because in my work, all the animals were not restricted to food excess, and that different from the previous study that before the experimental, the animal were fasted before, and they show something different. But in this case, they did not claim that the distinct behavior that changed from the pure control, from the pure condition. But in my case, I try to control everything in the regular environment and regular situation. So I can say that the energy balance or homeostatic system might be affect to the conditions reward. And the next thing that might affect to the condition reward is a macronutrient ingredients in the chocolate, because as I mentioned before, the portable food that is defined as a high sugar, high fat, and in the market, in the Thailand market, there are a lot of type of chocolate, and I use milky chocolate because there's high sugar, and high fat in mostly of them. That's why I use a milky chocolate. But that shows something different in the another research that they use a dark chocolate. Okay, and the last one is individual propensity. This factor, might be depends on their genetic background, because some work they found that on the same experimental design, they work with a rat. Some rat has a response to the chocolate, but some rat they no response. That's so I conclude that it might be the genetic background that induce them. And the last one, the conclusion. The environmental cue paired with the chocolate cannot activate the addictive-like behavior when animals were not restricted food access, and our findings supported that the evidence the chocolate cue might not be strong enough to trigger the reward process in the brain of the sated mice, that not strong to change the behavior pattern. Okay, I would like to thank you everyone for kind attention.
So, any question from the floor?
I would like to ask.
You can.
Yes, please.
Have you measured the weight of the rat in each group?
The way, yes, but I not present in this presentation. For during the experiment, that's maybe about 27 days. The weight not change significant. Yes, because during the condition they were they were given only 2 grams of chocolate per day. That's not much to change the weight.
So you said that the morphine-induced condition plus being blooded, it's something like an addict, right? Addict
Yes,
that cause
yes.
this.
Yes.
Yeah. But if we consider chocolate has some caffeine,
Yes.
as a component. And do you think that caffeine is also addict or not?
Caffeine does have a effect to addict also, but in case of chocolate, that's more complex more than only caffeine, because that have a lot of ingredient in the chocolate, also milk, sugar, and fat. That's I can't claim just only one ingredient that's affect to the conditions.
Yeah. My suggestion, if you use something other than chocolate, it would be less,
Yeah.
less conflict.
It might be good suggestion.
Okay. So, is there any more question from the floor? No? Okay. If there is no more question, yeah, I would like to thank you very much. And then,
Thank you very much also.
okay, thank you very much.